On December 2nd, 1942, an event took place in Chicago which was destined to change the lives of each one of us. This small group of men, working in deepest secrecy under the leadership of Enrico Fermi, achieved at this spot the first sustained nuclear reaction, and thus the power to produce neutrons in unlimited numbers. It is a matter of historical record that one of Fermi's first thoughts was the possibility of using these neutrons to prepare isotopes for use in medicine. It was only 2 years later, in a suburb of this same city, that Abbott began its studies with radioactive pentothal. Just why was such a substance an anesthetic? And how did it behave in the body? How could its action be further improved? Radioisotopes did indeed help provide the answer. This work also emphasized the possibility that there would soon open up an entirely new and virgin field of radiopharmaceuticals. Radioisotopes for medical use, as they come from a nuclear reactor, cyclotron, or radiochemical plant, require still further processing, purification, assay, and often conversion to proper pharmaceutical forms before they are ready for use. This has posed a challenge to those engaged in radiopharmaceutical work. The answer has been the development of many new handling techniques, new products, and new pharmaceutical forms. I-131 has assumed a commanding importance in medicine. It is currently produced only in the Oak Ridge reactor and converted to a solution of sodium radioiodide. A portion of this unrefined material is transferred as needed to the nearby Abbott radiopharmaceutical plant. This morning's supply of I-131, shielded with 200 pounds of lead, contains the equivalent of about 10 grams of radium. Radio gold, radio cobalt, and several other items were obtained at the same time. In the laboratory, one of several devoted exclusively to work with this particular isotope, the bottle of crude I-131 is moved to a heavily shielded but still easily accessible area inside a stainless steel hood. Aliquots of the solution are removed as needed for purification, the preparation of various pharmaceutical forms, or the synthesis of organic compounds. Extensive use is made of radioassays which are more rapid, more convenient, and more accurate than any chemical method. Assay of the gamma activity preferably is done with the Lorentz electroscope, in which the operator times the rate of the fall of a quartz fiber across a visual field. Beta rays are assayed in absolute terms using a Geiger counter and scaler, the activity being in the form of an infinitely thin evaporated sample. Aluminum absorbers are interposed to help establish the purity of the isotope. The assay sample is saved for measurement on many successive days to determine the half-life, again as a check on radiochemical purity. With all of these assays carried out on the material as received, and on every batch of every product made from it, the activity contained in any given volume, subsequently dispensed or packaged, can be stated accurately. Each radioisotope gives off a spectrum of rays of different energies. This complex electronic system automatically measures the complete gamma spectrum of each lot and provides a permanent graphic record of the radiochemical purity of the radiopharmaceuticals made from it. To establish the absence of other chemical forms of I-131, tests are made by means of paper chromatography. The sharp, intense peak and the absence of other peaks indicates that all of the activity is present as iodide. When all such tests show the desired degree of purity, the isotopes are released for further processing and use. Until recently, isotopes were handled and dispensed only in liquid form. Then came the idea of administering various radioisotopes as capsules. A method was developed in which the radioactive material is plated as an invisible layer on the interior wall of a high-strength gelatin capsule from which it cannot escape or cause contamination. To all appearances, such capsules are completely empty, Here is how the procedure is carried out. An accurately known amount of radioactivity, sufficient for many thousands of capsules, is purified and converted to a form from which it will deposit quantitatively in the capsule during processing. This is transferred in its shield to the encapsulating room. In the meantime, the gelatin capsules have been separated and the bodies placed in shielded trays. While 5 colors are used for activity identification, we shall follow today's production of red capsules, each to contain approximately 100 microcuries. The heart of the process is this specially designed machine. The operator wears 2 visual radiation detectors and a film badge, which provides a permanent record of the nominal radiation received, in spite of the heavy shielding of the instrument itself. A twist of the right wrist moves the capsules horizontally, and the foot motion lifts them into proper position to receive the isotope. Behind the leaded glass protection is a device which dispenses an accurately known amount of I-131 activity in carrier-free form into each capsule. After one row is filled, a movement of the left hand positions the next row for filling. The trays are then placed in this device, which completes the procedure whereby the material is caused to adhere firmly to the interior wall of the capsule. This unit is designed to produce several thousand capsules in a working day. Even though the capsule contains no liquid, each is tightly sealed so that, even if dropped, no harm or contamination will result. Each has been so treated as to resist even this bending test, which is far more strenuous than would be encountered in clinical use. For the first time in pharmaceutical practice, every capsule is individually assayed during production. This is only possible because the gamma ray activity permits assay from outside the capsule without damaging it in any way. A robot counter takes each capsule from a shielded hopper, positions it in front of the scintillation counter, located behind one of the instrument panels, assays it within a tolerance of 5%, records the count, and places it in the shielded jar designed to receive capsules counting in that range. The robot, working day and night, is essential if all capsules made in 1 week are to be ready for shipment some 5 days in advance of the official calibration date. Capsules are taken at random from the jars at various stages of the run and subjected to quantitative assay. The counting system is purposely made similar to that employed in clinical practice. The observed values are 100, 100, 97, 102, and 98% of the standard. The end result of this week's effort is 5 jars of I-131 capsules ready for distribution. Each preparation made during any 5-week period is of a different color. This color coding prevents confusion both at the point of production and in the user's laboratory. Such capsules decay to approximately 1/2 their original strength in 8 days. Hence, a shipment frequently consists of 3 or more activities to be used on successive weeks, as they, in turn, reach the appropriate activity for administration. For the convenience of the user, they are pre-calibrated for Wednesday, following the day of shipment. Here, for instance, is a teletyped request from the isotope laboratory of William Beaumont Hospital in Royal Oak, Michigan, for 5 different items. Even though the order has arrived just before noon, all materials will be sent out that same evening by air express and will be in the hospital ready for use the next morning. Had the order originated in Geneva, Oslo, or Honolulu, not more than 20 hours additional would be required for the delivery of the urgently needed material. The teletype order is converted to precise laboratory and shipping directives. Assays are calculated for 24 hours after shipment, so that the correct activity will be present at the time of administration to the patient. In the production section, this young lady fills the order from stocks on hand. Each vial will contain an appropriate number of capsules of known activity. These 4 vials, shipped together, will care for all of the I-131 diagnostic cases to be handled in that hospital for the next 3 to 4 weeks. The application of the principle of color-coded capsules to therapeutic doses required several years of additional study and the preparation of an even purer I-131. In this case, the I-131 capsule is assayed at the moment of preparation. 3 strengths are prepared: 1, 3, and 5 millicuries, so that through the use of a suitable combination of capsules, the same flexibility of dosage is possible as with solutions. Here, because of the higher activity involved, a lead shield is required. In spite of this, the weight of the shipping box is still less than 4 pounds. The components of the order, namely filled boxes, invoices, and shipping documents, meet in a packaging room. As it emerges, each box passes in front of a scintillation counter, which measures the radiation emitted. Thus, the final package, though sealed, attests that it contains the proper amount of activity and meets all shipping requirements regarding exterior radiation. To minimize exposure of workers, portions of the packaging line are shielded, and the packages are transported along the ceiling to the truck waiting outside the door. The order, received just before noon, is ready by 3:30. All parts have been prepared, assayed, the waybills written. The order is on the way to the Knoxville airport for loading onto the first available plane. It will be in Detroit by 11 p.m., approximately 12 hours after receipt of the order. Similar operations have been proceeding simultaneously in North Chicago to prepare and dispatch the radiopharmaceuticals leaving from there for William Beaumont Hospital. This institution maintains a radioisotope department typical of those found in hospitals throughout the United States. It is capable of carrying out a wide range of diagnostic and therapeutic procedures. Some of these are to be found on this morning's worksheet. The first patient has an anemia of unknown etiology. To carry out the Shilling test, he is given a capsule of cobalt-60 labeled vitamin B12. 2 hours later, a 1 milligram dose of inactive B12 is injected to ensure maximal excretion of the activity in the urine, and 24-hour specimens collected. Activity in a 1,000 cubic centimeter sample may be determined easily by setting the bottle containing the urine directly in contact with the crystal of a thoroughly shielded probe scintillator. The observed output of 4% is indicative of poor absorption. The test is repeated, now including, in addition, a capsule of intrinsic factor. The new output is found to be 12%, which is in the normal range, providing a highly probable diagnosis of pernicious anemia. This test, being independent of the patient's previous therapy, provides an easy means of securing a conclusive diagnosis. A second diagnostic procedure makes use of the I-131 derivative of a chemically pure fat, triolean. This material is available as capsules embedded in an oil-absorbent medium. Capsules may also be prepared just prior to use by adding to each an amount of tagged fat equivalent to 25 to 50 microcuries. At the same time, an identical amount is measured into a plastic-lined stool collection container in which have been placed 100 to 200 cubic centimeters of oil to simulate stool volume. This acts as the standard. The capsule is administered and the stools collected in 24-hour units. Counting is carried on from outside without opening the cartons. In this instance, a 48-hour output of 20% was found, well above the normal range of 2 to 3%. The test is repeated, using this time an iodine labeled oleic acid, which does not require pancreatic enzymes for its absorption. The lower value of 5% obtained on repeating the test suggests the presence of pancreatic disease. Thyroid studies with I-131 are the most common of all isotopic procedures. As is customary, the clinician interviews the patient, establishes that there are no contraindications, and decides that she is a suitable candidate. At the laboratory, she is given a capsule containing approximately 25 microcuries of sodium radioiodide. This is taken like any other capsule. A notation is made in the hospital records, and instructions are given to report at a definite time. It has been shown that absorption from such a capsule is more rapid than if a solution had been given. If the patient had been from an area some distance away, it would have been customary for the physician to give the capsule to the patient to be taken at home at the appointed time. The patient is seated in a chair with her head firmly positioned. A probe scintillator connected to a suitable scaler is placed at an accurately established distance, in this case, 20 centimeters from the front of the patient's neck. A count of approximately 2 minutes is made. The direct radiation from the thyroid is eliminated by covering the latter with a 1/2 inch thick B shield. A repeat count, therefore, measures the body background only. This is subtracted from the initial reading to secure the true thyroid count. A suitable phantom is needed to simulate the absorption and backscattering in the neck. This one was developed recently by the Oak Ridge Institute of Nuclear Studies. A capsule from the same lot, and therefore the same color as that given the patient, is placed in the phantom. A 2-minute count again is made. If the B shield is now swung into place over the phantom, we secure, by subtraction, the true phantom count. The thyroid count, divided by the phantom count, gives us directly the percent of the administered dose in the thyroid at that time. By convention, this is known as the thyroid uptake. The time after administration selected for the first measurement is usually between 2 and 8 hours. This study is particularly valuable in detecting hyperthyroidism. If there is any doubt as to the diagnosis, or if a therapeutic dose is to be given, the 24-hour thyroid activity is again measured in this same way. Absorption is usually maximal by this time. Many clinicians prefer to determine, in addition, the urinary output, the protein-bound iodine conversion ratio, and to carry out a scanning procedure as auxiliary tests to aid in achieving a correct diagnosis. It is to be emphasized that the thyroid uptake in itself is but one part of a broad spectrum of valuable tests, all available if I-131 is used as a diagnostic agent. The assembled data is discussed by the radioisotope director and the clinician, who decide upon what further steps may be desirable. Diagnosis in this case is hyperthyroidism, and it is agreed that a therapeutic dose of 6 millicuries of I-131 will be administered in capsule form. These 2 therapeutic capsules, taken together, contain the prescribed dose. They are administered in exactly the same way as the diagnostic capsules. The final case not only makes use of I-131, but provides an excellent illustration of the fact that I-131 often localizes in aberrant and metastatic lesions. Scanning with a Geiger-Muller tube directly in contact with the skin shows a significantly increased count over a circumscribed area in the left scapula and demonstrates that the lesion is of thyroid origin. Such a diagnosis and localization would not have been possible by any other means. A mechanical scanner may be used to make a pictorial record for the use of the surgeon or for further comparison after treatment. It is decided that the preferred mode of treatment is the oral administration of 50 millicuries of sodium radioiodide. All operations are carried out in a stainless steel tray. The shield cover is removed with a long mechanical finger. The bottle cap is grasped and unscrewed with a shielded device. The patient drinks directly from the bottle through a straw. A stream of water ensures the taking of the entire dose. In this way, the entire procedure may be carried out without significant exposure to operator or attendant personnel. Simplified techniques and instrumentation such as those which we have seen, together with the availability of diagnostic and therapeutic radioisotopes in easily employed pharmaceutical forms, are rapidly making isotopic procedures available to physicians trained in the use of these important new tools of medicine.